Right, question four. Question four is a little bit of a sneaky one because it's getting you to combine two different units, basically. It's getting you to think about your whole respiration unit and the unit you had on the nitrogen cycle, and it's combining those both. So it starts with carbon dioxide and atmospheric nitrogen. So gas and gas can be fixed by living organisms, and it's asking you for the definition of fixed. If you were to think, okay, fix it into a liquid, into a solid, that's not a bad start to the thought process. It does need to be any organic molecule that you have got, anything that is bioavailable, fixed in the living organism. So what could you fix carbon dioxide into? Any sugar, starch, carbohydrate, any of that. Atmospheric nitrogen, anything that's nitrogen based, whether that's a protein, whether it's ammonium. So literally fix it in a organic compound, so something that can be used in the living organism as an organic compound, or describe. I can have any named organic compounds there. Cool. Then the diagram here, if you look at it closely, the first time you're going to think, oh, I can see my electron transport chain here. If you look at that bit here, you've got your 3 proton pumps and you've got your ATP synthase. So if you're clicking, oh, that does look familiar from the whole electron transport chain, whether that's in photosynthesis or whether that's in respiration, it's irrelevant, but it looks like an electron transport chain. You've got a build up of that chemiosmotic gradient here. And yep, you've got some sort of electron acceptor. The electron seems to come from somewhere through the pumps and then accepted by, oh, not oxygen. Okay, so something else is accepting it. That bit here should look familiar from the nitrogen cycle. One of you has asked me just before the exam to go through the nitrogen cycle quickly. So that stuff was on the board literally until the second you started the exam. So the actual questions. Initially, whenever you get your diagram, understand your diagram first and see what actually is in that. What does it actually ask you? Name the nitrifying bacteria. That's 1 mark out of your 3. And describe how they produce a source of electrons. So naming the bacteria, and it says nitrifying, not nitrogen fixing. You can't give me Azotobacter, you can't give me the Rhizobium. You will have to give me the Nitrosomonas and the Nitrobacter. The order is irrelevant. If you kind of paid attention, Nitrosomonas, Monday monas comes first, and the Nitrobacter, because it's at the back, is your second one. And that's literally your first one is your Nitrosomonas, and that's your Nitrobacter equation there, literally. So that's the naming. And then how do they produce a source of electrons? Here we go. Here are the electrons made. Okay, you've got in this place here, you've got the electrons kind of not made. Made is the wrong word. You can't create matter. Well, you could, but not in biology, not in a biological context. So the electron is being released from where? The ammonium here is being turned into nitrite. That process released the electron in the Nitrosomonas. In the Nitrobacter, your nitrite is further then reacted with water to get your nitrate, and the electron is released in that step. So it's literally putting what is a chemical equation into words. That is what it's expecting you to do. So you're naming Nitrosomonas and your Nitrobacter, and then ammonium into nitrites and releasing it there. Or you can, for the nitrites, obviously the Nitrobacter, the nitrite into nitrates. We can refer to water as releasing the hydrogen and the electron together. Okay, right, back to the question. State, here we go. State the name given to those series of molecules. Here we go. The electrons are passed along a series of molecules in the membrane. Those molecules seem to, as per picture, they are shoving the protons from one side to the other. They're your proton pumps. What do we call the 3 proton pumps together? I got proton pumps from some of you, and I can't give it to you because it's about the series of molecules, not one proton pump by itself. All together, they are now called the electron transport chain. So that is needing to be there. I could have accepted electron carriers, but not just one proton pump, because it's about the series of molecules. How is the energy released from the movement of the electrons? Okay, so describe how the energy released from the movement of these electrons. Where do they move? Look at that in the diagram. Where do they move? The electrons are moving through those 3 proton pumps. What does that do? The energy is powering those proton pumps. It's giving them the energy to then move the protons into that space, which is here, the bacterial cell wall, not the intermembrane space, not the inside of the thylakoid, because it's not photosynthesis, it's not respiration, it's a process that those bacteria do. So where do the protons move? Move into the bacterial cell wall. Why? So we get the chemiosmotic gradient that then can be released through your ATP synthase, and then we get the production of ATP. So what do the electrons do? They're pumping protons against the gradient into the cell wall, creating that chemiosmotic gradient, building that up. Do you know what? I'm happy it's only 3 marks, because I don't think I would have moved, I would have done the against the gradient there myself. Okay, right. What is the next one here? The next one here. Describe how ADP is phosphorylated as a result of the reaction shown. You've got the chemiosmotic gradient. You need to, somewhere in your answer, have your ATP synthase. I've got from some of you beautifully 3 protons. You get a 120-degree spin, which physically pushes your phosphate into your ADP to then as a kind of condensation reaction to produce the ATP. So I've got that beautifully with far more detail than is actually needed. So the protons flow down the concentration gradient through that stalked particle ATP synthase. That provides energy to join the ADP and the phosphate together. Cool. Right, then this one here, because we can, it's now branching out and it's flicking over from what was a question on respiration or sort of electron transport chain and nitrogen. It now moves over into photosynthesis, just because it's another biochemical process, all the small scale stuff in one question. Nitrifying bacteria are chemoautotrophs. From your knowledge of the light-dependent reactions of photosynthesis in the Calvin cycle, so from what you already know, suggest how bacteria use the products of the reaction shown. Step 1, what products are actually shown? It doesn't say identify the products shown, but that will kind of give you the where do you need to go. Here we go. Here are your products. ATP is a product, and your NADH is shown here. So your reduced NAD. These ones are your products. So it's not NADPH, that's for plants. So for bacteria, it seems to just be reduced NAD that they are producing. So these ones are your products. What do you know about those products from the light independent part of photosynthesis? So these products come and they are then doing the carbon fixing. In photosynthesis, you've got your ribisco and then, your ribulose bisphosphate, and ribisco is the enzyme that is shoving your 5-carbon and your 1-carbon sugar together to make your 6-carbon sugar that then breaks into your 2 trioses into your glyceride, into glycerate phosphate. So here we go, GP. That fell out of my head there for a second. You've got your 3 carbon sugars made. Just the idea that those products are energy carriers, they are used in the carbon fixation in the photoautotrophs. So probably a similar process with the chemoautotrophs. So your GP are reduced to your triose phosphates by the NADH2 in plants. Using the energy from ATP, that is, the ATP is used to regenerate your rubisco. The carbon fixation part has to be there as a starting point. Okay, so there's a lot more detail in this one here.